In your book is a list of the most commonly tested autosomal dominant diseases. As a review, these are diseases that affect every generation, and an affected person always has an affected parent. I'll go through the key points for each one. Autosomal dominant polycystic kidney disease, or ADPKD, results in bilateral enlargement of the kidneys with multiple cysts, as you can see in this image. Patients present with flank pain, hematuria, hypertension, and progressive renal failure. 90% of cases are due to a mutation in the gene PKD1, and this is also associated with polycystic liver disease, Barrett aneurysms, and mitral valve prolapse. You can remember that this gene is found in chromosome 16 because there are 16 letters in the words polycystic kidney. Familial adenomatous polyposis is a condition in which the colon becomes covered with hundreds or thousands of adenomatous polyps, like the ones you can see here. Do you remember what adenomatous means? That refers to a benign epithelial tumor that arises from a gland. This occurs after puberty, which is common with many autosomal dominant diseases, and they almost always progress to colon cancer by age 40, if they're not resected. This disease is caused by deletion or mutation in the APC gene, which is on chromosome 5, which you can remember because there are 5 letters in the word polyp. The APC gene is part of the Wnt signaling pathway, which you'll learn more about in the reproduction lectures, and this mutation basically simulates constitutively active Wnt signaling, so it's kind of like the unusual growth years because the cells think that they're still in a developing embryo. Gardner syndrome, which is described in the GI chapter, is also caused by a mutation in the APC gene, but can be distinguished from familial adenomatous polyposis by the presence of osteomas, or bony growths, on the mandible or the skull. Familial hypercholesterolemia results in elevated LDL due to a defective or absent LDL receptor. Not having a functional LDL receptor prevents you from removing LDL from the blood, which means you end up with more LDL stuck in the blood. Since it's dominant, heterozygous individuals are affected, with a prevalence of about 1 in 500 individuals. And they often have a total serum cholesterol of over 300 milligrams per deciliter. Do you remember what normal cholesterol levels are? That would be between 140 and 250 milligrams per deciliter, although anything over 200 is considered borderline high. Homozygotes are very rare, but they have a much more severe form of this disease, with cholesterol levels often over 700 milligrams per deciliter. This can cause severe atherosclerosis even at early ages, which can result in myocardial infarctions before the age of 20. Patients may also present with tendon xanthomas, which are deposits of lipids, which you can see on this image on the hand, although most commonly they're in the Achilles tendon. Hereditary hemorrhagic telangiectasia is an inherited disorder of blood vessels which results in telangiectasias, which are dilated blood vessels near the surface of the skin. You can see a few in this image on the tongue. They can also cause recurrent epistaxis or nosebleeds, skin discolorations, and arteriovenous malformations or AVMs. Do you remember what AVMs are? They're abnormal connections between veins and arteries that allow blood to bypass capillaries and go directly from the artery to the vein, and they most commonly occur in the central nervous system. Hereditary spherocytosis results in sphere-shaped erythrocytes due to a defect in either spectrin or ankyrin. These are both proteins that maintain the structure and integrity of the plasma membrane. If they're mutated, this prevents the normal biconcave shape of red blood cells from forming, and therefore rather than seeing the normal central pallor in a red blood cell like you can see on this one, the cells appear uniform, like this one. These abnormally shaped red blood cells are destroyed in the spleen, resulting in hemolytic anemia. And it's diagnosed by looking for an increase in MCHC on lab tests, which is an indication of the concentration of hemoglobin in a given volume of erythrocytes. A splenectomy is curative because then the spherocytes won't be destroyed and won't cause problems. Huntington disease is a trinucleotide repeat disease caused by a CAG repeat on chromosome 4. You can remember this with the mnemonic hunting for food. This disease only affects patients once they reach a certain age, usually between 20 and 50. And in successive generations, it can have an earlier age of onset and more extreme symptoms. Do you remember what that's called? Right, anticipation. The major findings of Huntington disease include depression, progressive dementia, choreiform movements, and caudate atrophy, and also decreased GABA and acetylcholine in the brain. You can remember the caudate, acetylcholine, and GABA effects since they start with the letter CAG, which is the trinucleotide repeat that causes the disease. The caudate atrophy can be seen in this image as the lack of gray matter around the ventricles. As you can see, there's white matter right up against the ventricles all the way around. The choreiform movements are the most notorious, and they are unintentional, non-repetitive, rapid, jerky movements. In other words, the patient is unable to maintain a sustained posture. Marfan syndrome is a connective tissue disorder that mostly affects the skeleton, heart, and eyes, and results from a defect in the fibrillin gene. Findings include tall stature with long extremities and arachnodactyly, which means long tapering fingers and toes. Also, you'll see pectus excavatum, which is a concave chest like you can see here, and hyperextensive joints. These patients are also more likely to get systole-medial necrosis of the aorta, which you can see here, and which can result in aortic incompetence, a floppy mitral valve, and aortic dissection, which is bleeding into the wall of the aorta. They can also have subluxation, which means dislocation of the lens of the eye. Patients with Marfan's are also susceptible to Berry aneurysms. Can you think of a common location of Berry aneurysms? One of the most common is the circle of Willis. The multiple endocrine neoplasias include type 1, 2A, and 2B. The effects of these will be covered in more depth in the endocrine chapter, but for now, just know that they are genetic diseases that cause tumors of the endocrine glands, such as the pancreas, parathyroid, pituitary, thyroid, and adrenal medulla. Types 2A and 2B are caused by mutations in the RET gene. And mutations in this gene can also cause Hirschsprung disease. Type 1 is caused by mutation in the gene MEN1, which is a tumor suppressor gene. Now, if you think about it, this might seem weird that having a defect in a single copy of a gene causes a disease, since the other copy is still active. And remember, this is considered an autosomal dominant disease. Usually, if one copy of a gene is defective, the other one can still do its job just fine, and you have no disease. But remember, sometimes a spontaneous mutation can occur in an adult and shut down the other copy. With most genes, this is not a big deal because only that one single cell is affected. But if it's a tumor suppressor gene, it will only affect that one cell at first, but since it makes the cell neoplastic, you'll quickly end up with a lot of affected cells that all derive from the original one. Neurofibromatosis type 1, which is also called von Recklinghausen's disease, is a neurocutaneous disorder caused by a defect in the long arm of chromosome 17. Conveniently, there are 17 letters in the words von Recklinghausen, which you can use to remember this. Findings in this disease include cafe au lait spots, like this one, and neurocutaneous fibromas, like these. There are also neural tumors, such as optic gliomas, scoliosis, and Lisch nodules, which are pigmented iris hematobas, which you can see these brown spots here. Patients with NF1 are also more likely to get Wilm tumor, which are kidney tumors that typically occur in children. Neurofibromatosis type 2 is due to a defect in the NF2 gene, which is on chromosome 22. Again, this is convenient for memory since 22 is just two 2's. One of the most common and important findings in these patients is hearing loss, which is caused by bilateral acoustic schwannomas. Acoustic schwannomas are tumors of the myelin surrounding the 8th cranial nerve, which is required for hearing, as you can see in this image. These patients also get cataracts at a very young age. Tuberous sclerosis is a neurocutaneous disorder with incomplete penetrance and variable presentation. Do you remember what those mean? Incomplete penetrance is when not everyone with a mutant allele will have the disease, and variable expressivity is when people with the same mutant allele might have different disease traits. Tuberous sclerosis is pretty rare, occurring in only about 1 in 10,000 people. Just like multiple endocrine neoplasia, This is caused by a defect in a tumor suppressor gene, which in this case is the gene TSC2. This is why you have a variable presentation and incomplete penetrance, because if the healthy copy never gets mutated, then you'll never have any symptoms, and if it does get mutated, then the symptoms will depend on which cell type the second hit was in. Some symptoms you might see are tumors like adenomas, hamartomas, angiomyolipomas, rhabdomyomas, and astrocytomas, which in turn can cause other symptoms. So for example, an astrocytoma might cause seizures. In this picture here, we can see hypodense spots in the kidneys. And what type of tissue is hypodense? Adipose. So these are renal angiomyolipomas, which are benign tumors made of smooth muscle, blood vessels, and adipocytes. I know tuberous sclerosis might seem like a hard one to wrap your head around, but these renal angiomyolipomas are a good characteristic to remember, since you pretty much only see these with tuberous sclerosis. And if you have tuberous sclerosis, you almost always have them. The last autosomal dominant disease I'll cover is von Hippel-Lindau disease. This is also a neurocutaneous disorder, and is usually caused by deletion of the VHL tumor suppressor gene in chromosome 3, which you can remember since there are 3 words in von Hippel-Lindau. Now I talked about ubiquitin in another section, but to refresh your memory, ubiquitin is added to a protein that needs to be degraded. Like in this case it might be Huntingtin. The protein's aggregation causes Huntington disease. And ubiquitin targets it to the proteasome to be degraded. VHL normally works as part of the ubiquitin ligase machinery, so it helps to add ubiquitin to proteins to target them for degradation. When VHL is deleted or mutated, its target remains functional. Now the most important target in this disease is HIF, or hypoxia-inducible factor. As its name suggests, HIF is induced by hypoxia. Now what do you think a tissue might do as a response to hypoxia? Well, it makes new blood vessels to supply oxygen, which is called angiogenesis. If this happens in an uncontrolled manner, which is what's going on in von Hippel-Lindau disease since HIF can't be degraded, this leads to benign tumors of blood vessels called hemangioblastomas, which most commonly are found in the retina, cerebellum, and medulla. They can also cause bilateral renal cell carcinomas and some other tumors. In this image, you can see a cerebellar hemangioblastoma right here. Okay, let's do another flash quiz. I know we already did a few family trees, but more practice is always good. So what is the mode of inheritance of this trait? Remember, orange is someone who has the trait, and females are circles and males are squares. This one is mitochondrial, and the way we know this is again process of elimination. It's not autosomal recessive because it's in all 3 generations, and it's not X-linked recessive because it's mostly in males. It also can't be X-linked dominant because then the affected male would give it to all of his daughters, and he doesn't, which we can see here. Now technically this could be either autosomal dominant or mitochondrial, but how can you tell if a trait is mitochondrial? It's because an affected woman gives it to all of her offspring, and men don't pass it to anybody. And that's exactly what we see here, where this woman gives it to all of her children, and so does this one, but nobody else gets it from anybody else. Therefore, this one's mitochondrial inheritance.